Good morning. It's September 12th, and this is your daily brief in science. Here's everything you need to know. NanoViricides is advancing a broad-spectrum antiviral drug candidate called NV-387, which targets respiratory viruses by mimicking sulfated proteoglycans that viruses attach to on cells. This design makes NV-387 resistant to viral escape, which is a common challenge with traditional antivirals. Success in treating Mpox with NV-387 could lead to FDA approval as a smallpox therapeutic, opening the door to government and international stockpiling contracts worth over $10 billion. The drug's broad activity means it could reduce the need for frequent vaccine updates caused by viral mutations, potentially saving billions annually and enabling immediate treatment upon infection. Currently, NV-387 is moving toward Phase 2 trials for Mpox, supported by animal data showing effectiveness against respiratory viruses like RSV, influenza A, coronaviruses, as well as smallpox and measles. In animal models, NV-387 has achieved complete cures for RSV and demonstrated high efficacy in lethal models of Mpox and measles, both significant health threats with no approved treatments. NanoViricides plans to present at the LSX World Congress 2025 in Boston on September 17th, where leadership will discuss their drug pipeline, licensing opportunities, and the potential of broad-spectrum antivirals based on nanotechnology. The LSX Congress is a key event for biotech and pharma leaders to network and showcase innovations taking place at the Boston Convention Center. The presentation will highlight licensing strategies, market opportunities, and the revolutionary potential of nanomaterials to overcome viral resistance, enable drug rescue, and allow targeted tissue-specific delivery. Beyond NV-387, the company is developing treatments for herpes, viral eye diseases, and other pathogens, with future licensing plans for RSV, pox viruses, and enteroviruses. Their technology is licensed from TheraCore Pharma, providing broad rights to develop antivirals against diseases including HIV, hepatitis, rabies, Ebola, and various influenza strains. Additional candidates like NVHHV1 for herpes viruses and NVHIV1 are designed to prevent viral escape by targeting conserved entry sites, with efficacy demonstrated in animal models. LivingPhoenix has announced a significant advancement in collagen technology with its Pigment collagen. This collagen has been peer-reviewed and published in the Q1 journal Frontiers in Bioengineering and Biotechnology, confirming its scientific credibility. The company has established a detailed classification system for Pigment collagen, supporting its use in medical aesthetics, tissue engineering, and biomedical materials, with ongoing registration and approval efforts. Pigment collagen is filed with China's NMPA and has been incorporated into official standards, indicating its potential for clinical and industrial applications. LivingPhoenix holds multiple patents, including 3 material patents and 1 use patent granted, with additional patents filed internationally, and has registered in markets such as the US, EU, UK, South Korea, and Japan. The collagen technology originated from Kyoto University, was introduced to China by Japanese scientists, and has been locally upgraded in Chengdu, resulting in a second-generation independent IP system developed by a global team of PhDs. This collaboration between Chinese and Japanese scientists highlights China's growing capabilities in biomedical innovation through multiple patent filings and international efforts. The technology addresses a knowledge gap in collagen fiber self-assembly, supporting developments in tissue regeneration, anti-aging, and wound repair, backed by 4 Chinese invention patents. Structural insights from advanced cryo-EM visualization of collagen fibers provide a scientific basis for industrialization and clinical use, tackling key challenges in the field. Market forecasts project China's biomimetic collagen sector will grow rapidly from under 1 million yuan in 2025 to over 6.7 billion yuan by 2030, with a compound annual growth rate exceeding 150%, and Pigment collagen has been incorporated into national standards. The company has achieved mass production with an annual capacity of 30 tons, aiming to reduce costs and compete with recombinant collagen. A breakthrough collaboration with Qianfu Jincheng Laboratory, utilizing cryo-EM technology, has provided high-resolution structural insights into collagen fiber self-assembly, advancing the scientific understanding in this area. Octopus arms are highly adaptable, capable of complex behaviors like locomotion, probing, and object manipulation. These abilities are thanks to approximately 100 chemotactile suckers that function like a combination of nose, lips, and tongue. All 8 arms can perform a variety of actions, including shortening, elongating, bending, and twisting, demonstrating their remarkable flexibility. Multiple arm actions can occur simultaneously on the same or adjacent arms, indicating advanced coordination. Researchers have identified 12 distinct types of arm motions, with specific regions of each arm specialized for different actions, such as elongation at the base and bending at the tip. While all parts of an arm can perform all movement types, some motions are more common in certain regions, showing a hierarchical use of limbs. The study reveals a functional division where front arms mainly explore and grasp, while rear arms support movement like stilt and roll. Octopuses tend to use their front arms more for reaching and grasping, with about 61% of arm use occurring in the front and about 39% in the rear. Recent field research shows that their sensory-rich, flexible arms inspire the development of soft robots for search, rescue, and medical tasks in confined or hazardous environments. These findings enhance understanding of octopus biology and have implications for neuroscience, animal behavior, and bio-inspired robotics. Notably, octopuses do not favor right or left arms, but adapt limb use based on specific behaviors. They spend roughly 80% of their time in dens, emerging mainly to forage, which makes observing their natural movements challenging due to camouflage and habitat complexity. Their arms are composed of 4 muscle groups around a central nerve, enabling extreme flexibility without bones, allowing for complex movements. 3 scientists, Dr. Michael Welsh, Paul Negulescu, and Jesus Tito Gonzalez, have been awarded the 2025 Lasker-DeBakey Clinical Medical Research Award for their work on Trikafta, a triple drug therapy that has revolutionized cystic fibrosis treatment. Approved in 2019, Trikafta targets most CF cases by addressing the underlying defect in the CFTR protein, leading to a dramatic reduction in hospitalizations and lung transplants. This therapy has transformed cystic fibrosis from a fatal disease into a manageable condition for over 90% of patients, with many children now living healthier, more active lives. The team utilized innovative techniques, including dye-based chemical screening and real-time ion flow tracking systems co-invented by Gonzalez, to identify molecules capable of correcting CFTR malfunction. Research from Welsh's lab in the 1980s and 1990s revealed that the delta F508 mutation prevents CFTR proteins from reaching the cell surface, impairing essential ion flow. Prior treatments mainly manage symptoms rather than addressing the root cause of CF, which is caused by genetic mutations leading to mucus buildup in organs. Despite its success, challenges remain, such as high costs, around $300,000 annually, and limited effectiveness for some mutations, though efforts are underway to improve access. Vertex has launched a pilot donation program across 14 countries to help low- and middle-income populations access Trikafta. Negulescu emphasizes that understanding disease mechanisms at the molecular level is crucial for developing targeted therapies. Their discovery showed that the defective CFTR protein is not entirely broken, but trapped or malfunctioning, allowing drugs to restore its mobility and function. Gonzalez's co-invented ion flow tracking system accelerated drug screening and was vital in discovering effective CF modulators. Since its approval, Trikafta has extended life expectancy, with patients now living near normal lifespans if treatment begins early, compared to early childhood deaths in the past. A new study published in JAMA suggests that contrast-free biparametric MRI is just as effective as the standard multiparametric MRI in detecting prostate cancer. This could lead to major changes in screening guidelines worldwide. The research supports a shift toward faster, less resource-intensive imaging methods that do not rely on contrast agents. Eliminating dynamic contrast-enhanced sequences reduces scan times from 30 to 40 minutes to 15 to 20 minutes and removes risks associated with contrast dyes. It also cuts costs significantly, with the NHS saving nearly half and similar savings seen in the US and UK. The two-part MRI simplifies the procedure and could increase access to prostate scans globally. A large trial called PRIME, involving 555 men across 22 hospitals in 12 countries, confirmed that biparametric MRI matches the diagnostic accuracy of the traditional method. Experts highlight the potential for this approach to improve efficiency, safety, and affordability in prostate cancer diagnosis. However, they emphasize the need for standardization in imaging quality using tools like the Prostate Imaging Quality Scoring System. These findings come as the TRANSFORM trial, set to start later this year, aims to establish more effective screening protocols. Overall, this advancement could make prostate cancer detection more accessible and cost-effective, addressing the rising global demand for scans. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.